Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I'd like to thank all of you who joined us live at our first Dr. GPCR virtual cafe held on March 26th. Our next virtual cafe will be on April 29th at 11 a.m. EST. Our guest is Dr. Joanne Kamens, the Executive Director of AdGene. AdGene is dedicated to accelerating scientific discovery by enabling the easy, open sharing of high-quality controlled biomaterials, including those used in GPCR research. To reserve your free ticket, visit drgpcr.com/virtual-cafe today. To present your work or your company's technology, or to sponsor the Dr. GPCR Virtual Cafe edition, please fill out the form on the cafe's main page. The goal with the Virtual Cafe Talks is to bring you GPCR-focused live presentations every month. You'll be able to join us live and interact with the speakers and attendees. We hope you'll join us on April 29th. To stay on top of all your GPCR news, subscribe today to our monthly newsletter at drgpcr.com/newsletter. Last but not least, find us on YouTube and subscribe to our channel. On our channel, you can watch the latest Dr. GPCR podcast episodes and even the Virtual Cafe Talks. Come check out Dr. Brian Airy's presentation about GPCRs that are involved in modulating flow-induced signaling pathways in vascular endothelial cells. And now, let's dive into our episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I have the privilege of having with me Dr. David Gloriam from the University of Copenhagen at the Department of Drug Design and Pharmacology. Hi, David. How are you?
Hi, Jamina. How are you?
I'm great. Of course, I'm so happy that we could make it happen. Always happy to chat with you. So, how are you doing? It's been actually we spoke yesterday, so that the audience knows that we spoke briefly yesterday. But I wanted to touch base and ask you, how are you doing, and if you could tell us a little bit about your career and how you got into studying GPCRs.
Yeah, it's approaching Christmas time. We don't celebrate Thanksgiving here in Denmark, but you can feel that people are starting to get a bit more relaxed. It's a very nice time for me to look back, of course, and how it all started doing science. I think most people know me for maybe the database, GPCRDB, that about 4,000 researchers use every month. But my group actually does a lot of things. We do data science, analyzing receptor function, computational drug design, and we also started up the experimental lab for structural biology and have our first cryo-EM structure in-house.
That's great. So you do have that overarching ways of looking at GPCR structure and function relationships, which is really important. But when did you get into science? When did you get interested in science? And now I'm thinking, 14, 15-year-old or 20-year-old. Can you tell us about it, please?
Yeah, my first memory is actually when I was very young, a child. My grandmother died of cancer, and I felt that I would like to develop drugs when I became big. But these were, of course, one of many memories. My father worked in the chocolate factory, and of course, I worked in the chocolate factory too.
So you were David in the chocolate factory?
Yes, exactly. Yeah, exactly. I've always liked both, theory, but as well as practice, doing things with your hands, using your hands. So if you walk around my house, you'll see some furniture I built myself. But for this reason, I like chemistry. I could synthesize drugs, and I studied pharmaceutical sciences. Many people work in big pharma. But then I did a summer research group in my master's studies, and I got to try out science for 2 summers. And the first summer I did gene expression. I find it very interesting. I was very much into gene modification and therapies, gene therapy. But then in the second summer, I tested bioinformatics, and this blew me totally, because this was the time the first human genome had come. and we went on treasure hunt. So we identified actually in just a few years 26 new human receptors from the human genome sequence. So this was very exciting. And there was a lot of interest. So other groups, pharma, followed up on the receptors we identified. And actually the first open paper I published was the most downloaded from the journal from the whole year. So this was a very rewarding start of the science or the career.
And what did you feel like when you learned about computational biology, and did you feel like you were at home compared to being in the lab, for example? Or was it more stimulating for you?
In different ways. I mean, as I said from the beginning, I like working with the hands. I always thought it was very cool with all the different techniques and equipment in chemistry lab. And I still have a chemistry kit I do with the kids. But for me, another interest took over, which was analyzing data and planning the next experiments together with the team. And this is still what I think is most energizing and most fun. And as a bioinformatician, I could very quickly get a lot of data. And for me, that was more rewarding.
I guess it's mainly because it would get you to an answer faster when you have that computing capacity to analyze data faster instead of generating that data.
I have a lot of respect for all the work behind generating the data and pharmacologists that devote an entire lifetime or career to characterizing the receptor for the benefit of mankind, ultimately. But today, times are changing in the sense that whereas data generation used to be the limiting factor, today it's the analysis and the interpretation and understanding of the data, because we have so much data now, and it's actually our ability to understand it that's limiting.
Agreed. And we've come such a long way over the past 30 years with everything that comes to generating reliable data and acquiring data in high quantities compared to the 80s or even the 90s. So I think definitely there's a great complementarity between computational biology and the large amount of data that we can generate pretty quickly.
I think to the young listeners out there, it's a really exciting time now to get started on science because of all the data revolution there is.
Agreed. So you're at this point where you prefer computational work. How does that tie into GPCRs? When did you first hear about GPCRs?
Yeah. I think my work and preference has always been interdisciplinary, and it still is. So I like it when you can generate hypotheses on the computer, but then try it out experimentally. And I think that's when it gets the value. And that was also the environment I was trained in. So I first heard of GPCRs when I did the summer research school in my master's studies. And that was actually the Faculty of Medicine. There were a lot of pharmacology groups there. And they had, very nerdy pharmacology sessions in journal clubs. I was on the computer, but I was part of this environment. And it was very fruitful. And I think it was very useful later for being able to communicate with experimentalists. And everyone was passionate about receptors.
Absolutely. So you had, we were chatting and you had a really funny story about getting into studying GPCRs. Would you mind sharing that story with us? I'm really curious. Sometimes, depending on who I speak to, and even from my own experience, it's always either a class or a paper or a presentation that, you first read about the receptors and you just become fascinated with them.
Yes. I think I had a special interest for GPCRs because I knew they were really big drug targets. And I wanted to work with something that could either become a drug or the target of a drug. So I think that was the main reason why I got interested in GPCRs. And then, of course, I happened to be in a really exciting group in a really exciting time where we could discover all these new GPCRs from the human genome sequence. And I was in the group of Helgi Schioth. He's a rather tough group leader, But I learned a lot from this. I got a lot of collaborations, a lot of publications. And he invented, for example, the classification of GPCRs, the graphs nomenclature.
Wow. You're not the first guest who says that they had, a tough group leader, but they learned a lot. And I think there's an advantage sometimes when things are more difficult and you really have to work up to that challenge. That's when it becomes more productive at that point. And you also get to collaborate with a lot of people, which is phenomenal. So please go ahead.
Yeah. So it can be tough at the time, and I think it is tough to be a young scientist. But in retrospect, I'm thankful because it gave me exactly what I needed to promote my career. I got the publications, I got the grit, I got the sense for hard work, but also collaborative teamwork. And all of these were things that maybe I didn't see or understand at the moment, but I've come to appreciate afterwards. And actually, me and Helgi, about 2 years back, we published a new article together, a collaboration, where we looked at the trends in drug discovery on GPCRs. And actually, it's the most cited paper from my group, so it has more than 500 citations today.
Wow. Yeah, I feel like when you're a trainee, whether a PhD or a postdoc, and you get thrown into the deep end of the water, it might seem very overwhelming. But I feel like that's how you learn to swim. Mm-hmm. And in retrospect, I've had difficult times working in different labs, but at the end, I always came out with a positive because I learned more about myself. I pushed myself, and there were papers that came out, and got to know a lot of great scientists in the meantime. Mm. So getting back to GPCRs, since you're doing computational biology, do you have any favorite families or favorite receptors?
I used to. Because I discovered these new receptors. They used to be called orphan receptors. Like, orphan child is a child without parents. Orphan receptor is a receptor without a known endogenous ligand and function. And for some of these, all you knew was basically the protein sequence, nothing else. And because I think as most researchers, are driven by curiosity, I really wanted to understand the function better of these receptors, to see, could we discover a new physiological signaling system? Could they be relevant for drug discovery? So for many years, I focused on orphan receptors. And when I went to postdocs in UK, and so I acquired new methods. So I switched from bioinformatics to computational drug design. And I started developing tool compounds when I came to Copenhagen, Denmark. So I added more methods in my toolbox, but I continue to focus on orphan receptors, trying to identify tool compounds to find the functions.
That's fantastic. So any favorites in the orphan receptors that you worked on, that you actually discovered what their ligands were? Anything that stands out that you can tell us about? I'm really curious. I feel like orphan receptors, up until now, there are still orphan receptors. There's still a lot of work to be done. And they may or may not, but I think they do play an important role in pathophysiological settings.
Mm-hmm. Yeah, it's difficult to pick a favorite, but I'll try. So I think last year was a little bit of a climax for me, because I had been working on ERSD starting grant for 5 years, and then we discovered physiological hormones, peptide ligands that could activate orphan receptors and thereby potentially connect them to physiological function. So there were 5 receptors in there. We actually aimed in the application to de-orphanize 1 or 2 receptors. And usually you don't live up to your aims, but here we found more than we thought. When one of these GPR55, it had a subnanomolar potency. So that was very interesting. And that was actually known ligand backup from a class B receptor. So that was very intriguing. How does that work? Why is that? But then there's also another receptor. One of the receptors I discovered in my PhD, GPR139, where in my group, we first identified several tool compounds, and then we published proposed endogenous ligands, amino acids or peptides. So potentially it could have been de-orphanized now. And through a collaboration also, there is a group now that has the structure of that receptor. where my group has contributed to the design of the constructs. So that's a full circle, and that is very fun for me to start with the identification of receptor, to go all the way to the functional characterization and eventually a structure that is the best template for rational drug design.
Agreed. Can you walk us through, the whole full circle that you just mentioned, from the moment you find a sequence to the moment where you actually get to a point where you do have a ligand and you're kind of de-orphanizing and better understanding the physiological context in which this receptor works? Really basic. And how does this process involve different groups?
Oh yeah, it's a really good case story, I think, for the different disciplines. Because initially, when we first discovered the receptors, what we could do, we knew very little. We could do sequence-based phylogenetic trees to show the relationships to other receptors. We could mine databases for mRNA to look at tissue expression, but that's limited data. And then later we could use virtual screening on the computer to pick some small molecules that maybe would be active in the lab. And eventually they were assayed by pharmacologists, and we found some real tool compounds. And for the physiological hormones, we'd be using machine learning and genomic analysis. And then, of course, for the structure determination, structure biology collaborations. So it all comes nicely together. And then there are other projects where we follow up with animal studies also.
That's so fascinating, because now that you're describing it, it's really an interdisciplinary collaboration that gets you from the point where you discover a sequence to the point where you actually understand what the implication of that sequence is at the protein level, which is really fascinating. So you mentioned that you'd do some virtual screening, but also you'd look at different mRNA databases. How reliable are those that information that's outside, for example, in a database?
Well, I think a prediction is always going to be a prediction. It's a best educated guess.
Yes.
It depends on the data you have. Fortunately, today we have a lot more data than we used to, both in terms of volume, but also in terms of different types of data. And I think one of the missions of the database GPCRDB is to integrate this data and make it more accessible to experimentalists. So, for example, we do a data-driven design of new experiments. So you can go online in your web browser and you can design mutagenetics experiments, or you can design constructs for crystallography based on the available data.
That's great. Let's take a step back to the GPCRDB. How did you come up with the idea, and how long did it take you, to at least get a first version out?
Oh yeah. First, I should give some credit to the people who started GPCRDB, Helgi and others. They started as a mail server in 1993. That was before internet existed, right? So that was great, and I have a lot of respect for all the beautiful work they did over the years. But then Hat was getting close to retirement. We met at conferences, and I was very much hoping I could maybe, if I was lucky, be the group who could carry on this hub. And actually, it happened. And that was one of the major single big decisions for me, because at the time I didn't have tenure. And the lead developer in my group, he didn't have a very long contract. I think we had like 2 years, each of us. But we can we really do this? It's a big responsibility for the community. But we also saw a really big opportunity. And we decided, let's give it a chance. And from that day, we've never regretted that. So the community response has been fantastic. When I walk into a conference, people will greet me that I've never met before, and they will say, "We love GPCRDB." So that's just fantastic.
So you went from David at the chocolate factory to David GPCRDB
Yeah,
at this point, which is definitely more career appropriate when you think of it.
yeah,
And how many people use it? What do people tell you when they say, "Hey, we love GPCRDB"?
yeah. Yeah, we have about actually on the main page of GPCRDB, you can see how many people used it in last month and the last year. So it varies slightly, but last time I checked, it was actually more than 4,400 in the last month unique users. And we've seen it steadily increased over the years, which I think shows it's more relevant for a wider audience.
Yeah, every time, to be honest, every time I have any uncertainties or any questions, that's the first place I go and take a look at the available information there, because it's really relevant and it's really easy to find. So I love the fact that you wonder something, you just go on there, and that's the starting point, and usually you get great answers to your questions out of there.
I've always liked actually to support research and support others. I think there are some researchers that are big egos, but I've always liked to, if I can help people, and I find it motivating and intriguing, just trying to make an interface that is easier to understand and more intuitive. And when people can use it and do something useful, then I'm happy. Then I feel I've accomplished something. So I'm not always the person who needs to get the final result. If I can be the person who develops the tool, I'll be happy.
Of course. And I saw at the latest GDR meeting that you had given a talk about GPCRDB and shown the different, landing pages that changed over the years. Is that how do I put the question? Is that something that you're continuing to develop over time? So you're focusing on the looks of and the usability of the database, but also on adding information in there.
Yeah, most certainly. So we've actually extended the section where you can give feedback to GPCRDB. So you can submit, requests and wishes for new tools, but also improvements of the existing and interface and anything. And when we publish a new tool online or in a paper, we always put a lot of care in making sure that it's clear and can be understood, because we don't want to use 3 years to develop something and then people don't know how to use it. So typically we ask people around us to give comments and try it out. Maybe some people from the lab.
That's great. So I'm putting this out to the audience as well. Please go take a look at GPCRDB. If you have any comments, any input, I think David and the team are going to be very happy to hear from you, because it's a tool not for one group, it's a tool for everyone in the community. And the goal is to really better understand GPCR function and structure relationships. Speaking of that, what do you think are the challenges that should be addressed to facilitate the understanding of function of receptors?
Yeah.
It's a tough one.
No, yeah, well, I have two answers. I'm just thinking about which to start with. So, as having had a group for some time, I am thinking about what are the big research challenges where my group can do something unique. And maybe I'd like to apply for a big center, that runs for many years and achieve something bigger than previously. And what I think there's two main areas that interest me. And one is understanding receptor function on the single residue level. So I like to see this as data accelerated receptor evolution. So acronym there. So, evolution have optimized the receptor proteins for many millions of years. But I think with computational methods today, we can actually simulate more interactions than nature have done on a computer much quicker than been done in a million years. And if we could understand the determinants of expression and signaling and binding and on the molecular level, we should also be able to engineer receptors for specific purposes or engineer, for example, peptide ligands become drugs. So that is something that interests me, the fundamental mechanisms down to the residue level. And the other thing is the challenge of safer drugs by biased signaling, where a drug was approved from Trevena, Lisridine recently, but it's been questioned whether it actually is biased or just a partial agonist. Setting aside the definition of that, the main challenge remains, which is to characterize all the pathways so you know which pathway to target, which pathway gives the therapeutic effect and which one gives the side effect, but then also to develop all the drugs that can give that selective response.
Agreed. I think having having the tools to better understand at the residue level how that influences receptor function or how that influences ligand action on a specific receptor is definitely a great way to really dig deep into what's going on. But then yes, you do also need the systems to make sure that you can test those compounds, for example. And at the end of the day, if the goal is to treat a disease, you need to understand where things have gone wrong. And it's most likely not one pathway, it's maybe the accumulation of multiple pathways. So having that ability to readjust the entire ship instead of going at it with, as someone put it yesterday, with a hammer, with one way of doing this. So I know we touched upon this in the beginning, and we've been kind of talking about it, but how do you think, in the larger sense, computational work and machine learning can help GPCR research, the big picture?
Yeah, I think it already has. There's been drugs discovered based on computational methods. We've learned a lot about the dynamics of receptors using computer simulations. I think going forward, as I mentioned in the beginning, times have changed where data science will become increasingly important because we'll have the bottleneck will be analyzing and understanding the data that exists, not generation of data. And for me, using all the developments, for example, in machine learning, to do something useful, understand the receptor function on the residue level, and use this and apply this in drug discovery or in other experiments.
You mentioned that, in the beginning, you had about 2 years to you didn't have tenure, you had 2 years to assert yourself, and then you decided to go into the GPCRDB. How has your group grown since then? How big is your group at the moment?
Yeah, currently it's about 15 people in the group. I'm also fortunate to lead what is called the research cluster at my department, consisting of 8 groups doing pharmacology, chemistry, structural biology. So that's really nice. When I started out, I had a relatively small group for a while. Then I was lucky enough to get a European starting grant. And I was actually so lucky that I got a Danish grant at the same time. So I'm not from Denmark originally, I'm from Sweden, but one of the reasons I stayed in Denmark is there's some really good foundations here that support research. So I got 2 group starting grants, and then of course my group grow. But then what's happened in the last years is I've had more senior people in the group. So whereas in the beginning I had maybe a majority of PhD students and master students and a few postdocs, today I have more postdocs and even assistant professors in the group. And for me, that helps me to do higher level research, because when I come to office, I'm not just the supervisor, having all the right answers. When I go to work, I meet in with colleagues on a really high level, and I find that we have a really good sparring. It's really energizing. And I feel that many of the ideas for new projects or solutions to problems comes from them. And that's actually what makes me the most proud. When I come into work and I have a group member that does something better than I do.
I think we've touched upon this in a previous conversation that we had, and it really resonated with me. So you're at a point now where you surrounded yourself, or you have a group where people are actually doing research at a high level. Not that, having trainees is a bad thing, but if you want to advance, you need, and it comes with time, you need a little bit more experience, and having that ability of having such a large group, I think is really conductive to doing big things and to getting answers maybe faster. So how do your days go by? Well, now we're in a COVID situation, but let's say it's a typical year. It's 2019. It's not 2020. What kind of interactions do you usually have with your team, and how do you make sure that your team knows that, for example, you're there to support them?
Yeah, being there to support them, that's the key, right? When I had a small group, I would start in the morning, walking around the offices and asking people if they had what they needed in person. And if they didn't, I would sit down with them and I would help them the time it takes. And then whatever I need to do, I do in the evenings or in the weekends.
Wow.
Today, I can't do that anymore. But at the same time, I don't want to give group members the experience that, the group leader is a bottleneck or not being present because it's too big of a group. And, I like research management or leadership, and I've taken some courses. And I also want to support the people who want to train as becoming group leaders. So what I did a few years back is I split my group in 4 subgroups. So each subgroup has a project manager, which is a senior researcher. And then that is the first go-to. So that would be the daily supervisor. And then we have meetings every 3rd week or so where everyone presents their project in the subgroup, and every member can help each other and learn from each other. And they have journal clubs and scientific talks.
Mm-hmm. What gave you the idea to split up your larger group into 4 smaller groups? And I have someone else did that, but in a completely different context. I'll let you answer the question first.
Yeah, well, it was a necessity. I had to. I found myself leading group meetings with 15 people around the table, where people felt they couldn't say everything they want to say because there wasn't the time, while other people were starting to pick up their mobile phones because what they heard was not relevant to them. And that just means it's time instead to split up to smaller groups where you have a common interest and you can help each other because you do similar work. At the same time, we do still have group meetings, but when we do, we focus on transferable skills, how to give a good presentation or something like that.
That's interesting. So the thought that came to mind when you mentioned that you were splitting the groups in 4 is actually, I forgot. The Virgin CEO, oh goodness, I forgot his name, Richard Branson, who every time he formed a company and the company reached, I think, 150 people or more, then he split it and made sure that each company is part of a smaller group. Because in his mind, the smaller the group, the better the communication, and it's easier. So it comes back to basically what you've said. And people get to know each other and work better together in these smaller groups.
Yeah, there has to be a room to grow. There has to be a room to express your ideas. And then if you should have critical mass, of course, but it shouldn't be too big either.
Yeah, I think sometimes it's and you had mentioned that, with 15 people, and I guess you had 1 or maybe an hour and a half of group meeting, which is definitely not enough to go around the table and make sure that everyone says what they need to say. Otherwise, it would be a 5-hour meeting, which no one enjoys.
Yeah. I still have an open door policy, and people can walk in my door anytime they want. And I still think it's a lot of fun, actually the most fun, to interact with members of the group. But I feel there are less bottlenecks, and I feel they don't have to. And I feel there are other people that can do things, and I can trust that will be done well.
Agreed on that. And we talked previously as well about, supporting professional development of the people in your group. So first of all, let me take a step back, actually. You mentioned that you took some leadership classes.
Yeah.
We know that in general in academia, it's not something that, professors have to do. What led you to think about taking these classes? Or to take them, actually? Was it again a need that you've seen that there was something not working in your group and you wanted to ameliorate, get it to a better working phase?
So I think the first time someone advised it to me was when I had some mentors and I was applying for a group starting grant, and they said that if you get a big group, the foundation will want to know that you will be able to lead it. And therefore, you should include funding for a research leadership course in your budget. And so I did. And actually, I took the course before I got the grant. So even as, I think I was a time-limited assistant professor at the time, I took my first research leadership course. And I didn't take the local course at my university. I took the national course. And you should do that. You should take the best possible course, not just the closest course. And that was extremely useful. So I learned a lot. And I would even say if I hadn't, I'm not sure I would have, survived the competition, because it's really competitive to get those group starting grants. And then later down the line, I've taken additional courses and I've chosen additional mentors. So one thing I did, I took coaching. So I had coaching sessions and I made my own agenda. So I showed up on the first meeting with my problems, and then I had these really professional people helping me to solve those problems and then to implement them together with group leaders. And that was really good. And then, as I mentioned, I also selected mentors. And what I've done is I reach out to a person that is a little bit older than me and have recently solved the challenge I have. And therefore, I can get tips about how to solve it myself.
I think that's fantastic. That's such a phenomenal way to marry, your scientific interests, your scientific abilities with the leadership abilities that we don't get training in when you're doing science, when you're working in the lab or you're working in front of a computer. And I think we should all have access to these classes so that we can become better leaders, better professors, better mentors. Speaking of mentors, for the audience, especially for the younger audience, how do you go about, and you've spoken to it a little bit about it, but how do you go about finding a mentor? Kind of like a protocol type of step-by-step. Because my guess is you won't go up to someone and say, hey, I really like your work. Would you be my mentor? What's the best way to get there?
Well, do a good job, and then people will see, and be passionate, and then people will feel that they want to invest in you. But I've contacted them maybe by email on the first case, but we know each other a little bit from before, have met before, and I'm very respectful. For example, of their time, and I don't take it for granted. They will say yes. If possible, I try to give something back, maybe mentor a person of their group or helping out with something. But I find that if you really respect their experience and tell them why you want to do this and why you select them, most people will say yes.
Agreed. I think based on my different conversations with previous podcast guests, with you as well, and with other colleagues, sometimes, especially with trainees, there is an inhibiting factor that says, well, I'm just a trainee. But I think if you, as you mentioned, if you respect other people's time and you give value, then you develop these wonderful relationships. And I know many people who said, I'm successful and I cannot wait to give back. And it's something that's not necessarily advertised, but not everyone has the time, not everyone has the desire to mentor, but those who do really are wonderful to learn from.
Yeah, I think it would be interesting to have more data and hear from many people, but I would guess that this is a common trait among many researchers, that they constantly are curious and want to learn new things. So lifelong learning. I mean, the day I stop learning, I might as well die. I mean, then it's so, for me, the actual development, that's the thing, and that's the fun part. And when I'm done, I get bored, and then I want to try something new. And I think that's why people choose to be researchers. No day is like the previous day.
Agreed. And I think someone once asked me, so how do you deal with problems? And I said there was no such thing as problems, just obstacles that you have to get through, new questions to work on. Speaking of continuous learning and moving ahead, what is exciting for you at the moment in the GPCR field? What is making you get up in the morning and say, I cannot wait to do this?
Mm-hmm. That's a big question.
It's a morning like this. I mean, please go ahead.
No, yeah. So I think there's a short-term answer, like a good day, and there's a long-term answer. Why do I keep doing this? Or why don't I do anything else? I think the short-term answer is, the fun being the sparring with the colleagues, the intellectual discussion, and looking at new data and interpreting that and planning the next experiment. That's a good day. Or seeing other people grow, seeing them develop and have a good time. On the long term, it's doing something that has an impact, something that's useful to others. And I do basic research, so I'm very far from, a clinical drug or anything. So maybe that's why I still build furniture at home. I can see a product. But if I can do something that's useful, and having been a group leader for some time, and not just thinking about survival, but starting to think about where I can have a real impact, and if I would get a center for 10 years, what do I want to do with 10 years of my life? How can I be most useful? Those type of things.
Agreed. And you mentioned, the excitement of, getting new data and analyzing data. And we talked a little bit about this, but what do you think are some tools or initiatives that could really speed up targeting GPCRs and in basic research, and better understanding GPCR function in basic research that can be translatable to in a disease setting, for example?
I think there's been many revolutions. I mean, as I mentioned, there was this treasure hunt with the human genome and identifying genes. And when I started my PhD, and people thought now we will be able to cure every disease, but we couldn't. And then there's been one thing after the other. The structure revolution in GPCRs, bias signaling. So for me, I think it's this harnessing these things and trying to implement them and tie them together. So, for example, could we develop safer drugs? Could bias signaling go from just theory to drugs? Can the computational models go from precise predictions to actually understanding receptor function on the residue level and be used to design more experiments?
Absolutely. So I always ask this just to almost all my guests. Do you think GPCRs are still a good drug target?
Yeah. So I think my group, we want to do high-level science. We want to understand receptor function with data analysis. We want to generate new structures and ligands and probes for functions. But then the GPCRDB database ties together with that. So we can develop tools to do science. And when we have science, we can deposit the data sets in GPCRDB. And that's something I like to see more of. More data coming into GPCRDB. And one dream I have is open science. So what if it could be a forum where people share their ideas, the big scientific challenges, they correspond on this, and then eventually they would solve big challenges together, and the data would be saved in a database. So for example, biosignaling, where it's a huge challenge to characterize all the pathways of all the GPCRs and their effects, and then to identify ligands that will selectively activate those pathways.
Yes, I think a lot of teams are working on that, including Michel Bouvier's team with all the biosensors, where I feel like it's in my mind, it looks like a big puzzle, and we're trying to piece the pieces together. And to do that, we need all these disciplines to come together and share ideas, share data. And I absolutely support the idea of this open science, where you can all put our heads together and get to better understanding these systems that we're looking at, and hopefully translate those into the clinic and help people with so many diseases. That any disease I can think of, it has something to do with the GPCR.
Yeah. So yeah, exactly. So if my dream comes through, then GPCRDB could be a hub for the community to solve these challenges, because really it is a community effort. And having said that, I want to also express my admiration for Dr. GPCR and what you are doing, because you are so good at the outreach, and it's going to have a huge impact. And I know also you have plans for scientific ideas. And I think you're doing a great job in being a forum for science.
Thank you. I really appreciate it. I think it started out with a very basic idea of just doing a podcast, because I wanted to talk to scientists, which I didn't really get a chance to do while working on the lab. I was always busy pipetting and, running experiments and making sure my cells were looking great for stimulation before an assay. But I think I've gotten a huge amount of comments from people saying it's a great program, and I very much enjoy doing it, and hope it does. Everybody feels how much I really enjoy talking to you and to other scientists, and it's my way of contributing to the field without a pipette. Although I loved pipetting as well, but I also always felt like it was not satisfying after a while, because it's the challenge. You were talking about challenges, and it was very challenging in the beginning. I learned how to do some techniques. I learned a lot, developed some tools, and took those tools up to the next level. But at some point, I needed an extra challenge, and I think the podcast and Dr. GPCR are my way of contributing to the field, of which I'm fond of. I think that by now everybody knows that I'm really fond of GPCRs.
And that's a unique contribution.
Thank you. I really appreciate it. The other thing I wanted to ask you about, so we talked a little bit about, how you went and got more leadership skills and how you manage your team, and it's a pretty huge team from various disciplines. What would be your advice for junior scientists who wanted to contribute in the field, whether in the lab or as a chemist or as a computational or machine learning scientist?
There's so many things. I actually gave a presentation recently to young scientists about the major challenges. I became a group leader and some tips. I would say work with the best, because really the research environment you're in has a huge impact, both on the thinking you develop and of course your opportunities to publish and get to where you want, the network. And don't be afraid to reach out. Go to as many conferences as you can, maybe online at the moment, but when you can, in person. Find out what motivates you, and play your strengths and have fun.
Agreed on the having fun and also going to conferences. Sometimes it can be intimidating for a junior scientist, to go to a conference and then sit with a Nobel Prize laureate or with a speaker. But we're all human. I think we all have the same interest of advancing the field. And if I can give one piece of advice, just go out there. Just go out there and talk to people. You'll see that, we all have the same goal and we want to all work together at this point. We've talked a little bit about science. We've talked a little bit about your favorite orphan GPCRs. Were there any moments in your trajectory as a scientist where you said, "Oh, aha, this changes how I think either about the field or how I think about the science that I'm contributing to"?
Yeah. When I think back, there can be some big moments. I can think of three, and when we decided to take over GPCRDB, that was one. Another one was when I was to the interview to get the first group starting grant, the European ERC starting grant, because I knew this would be decisive as to whether I would have a career or not, up to a single meeting. I prepared a lot. I had mock interviews. I traveled there a day beforehand and visited the place, and I took a course. And when I was there, of course, there was a waiting room, very nervous, of course. And I chatted a bit with the other people, but then I totally locked out that and focused on what I was going to do, and kind of chanted and memorized everything. I had 10 minutes to present all the questions. And this was the first time I bought, or the only time I bought a real expensive shirt, a brand shirt. And I think it's a Gucci shirt because I heard at the course one should have clothes in which one is comfortable, but also stands out. So you remembered. So I had this kind of orange-red shirt under my tuxedo, and I didn't want to have a tie because that was too much. But I found a shirt where there's something over the buttons because I didn't want to show the buttons. So that was just perfect. I bought this expensive shirt. I prepared, and I was 100% loaded. I walked in there, and I did the best performance ever. And the crowd just wow. And I walked out, and I felt, I think I did it. I did it. Yes, and I got the grant. So that was certainly a big moment for me. And I think the third big thing, I think I'd like to extend a big thanks to my partner because in my scientific career, I've moved 4 times for the job. And that's pretty easy to do if you're alone. But I mean, we timed the first pregnancy with a paternity leave during maternity leave during my postdoc. And we moved at least 2 times with kids. And now I think we're settled down, we're stable, but my wife has been very supportive throughout the process, and I try to be supportive back in all the ways I can. But hadn't she been willing to move, I wouldn't have had the same opportunities.
I'm happy you bring this up, and it's a question that I wanted also to ask you. How do you now, and how did you back in the day juggle, family life and work, and how do you do it? Sometimes, in Europe, usually people maybe have children as a PhD or as a postdoc. It's not that much the case in the US, but still having a family, having a partner where you have actually 2 careers to move is very difficult. How do you juggle with that?
Yeah. I have 2 kids, now 14 and 8. I have 8 months of documented paternity leave, and I had a bit more unpaid. So a key word is work-life balance. But a work-life balance is different for different people. For some people, that's a normal work week in terms of hours. For other people, it's a lot more. And I think I've learned that you have to be sustainable. You can't work hard unless you relax hard or whatever. Well, and I think if you're wise enough, over time you learn to listen to your body, and you learn to listen to the people around you, and how you thrive, how you do well, how you live well and how you perform well. So you find that balance. I've had a lot of hobbies, and for some time when research was taking more and more of my time, I felt some bad consciousness for not having time for those hobbies, or maybe for all the friends I used to have, childhood friends. Then I realized actually science is more fun. So I didn't need to feel bad about not having time for all the old hobbies, because it was more fun to analyze the data set and talk to my colleagues than to do inline skating. And today, I prioritize my family first. So if the kids need me, I'm the person with a flexible job, I'll be there. When COVID shut down the schools, I did homeschooling. And there will be days when my kids decide what to do. So, my daughter says, if we climb a tree, we climb a tree. If we draw a unicorn, we draw a unicorn. And that's very important, sacred family time.
I think so too. I feel like the way I think about it is it's also an experiment in life. It's not only an experiment in the lab or an experiment analyzing data on a computer, but also it's a matter of trial and error sometimes. And the way I see it also is it's a marathon. So it's not a 100-meter sprint, it's a marathon, and you need to find that balance. And if you can't find it in the beginning, just find new ways. I'll modify the parameters one at a time, ideally, until you find that pace that works well for you as a scientist, but also for you as a family, as someone who has a family, and especially when they're young. So as you know, I have a 5-year-old, and my priority is my 5-year-old. If we need to go down and as you mentioned, draw on the ground with chalk, then that's what we're going to do. Because in 5 or 10 years, he's not going to want to do that. So agree. There'll come a time when they'll need their independence, and we need to take advantage of this time when they're younger as much as possible. On that note, I have one last question. Where can people find you? And if you have jobs or openings in your team, where can people look for those?
So don't hesitate to contact me. Send me an email. Call me up. If you go into a conference and I'm there, say hi. And if you listen to this podcast, continue the discussion, whatever interests you and what is relevant for you. When we have jobs, we put on the website of the University of Copenhagen, where you find the jobs. We also announce them in Nature Jobs and Science Careers. If it's a programming job, we put in GPCRDB. I have an infrastructure grant pending for GPCRDB. If I get the grant, we will expand and we'll have all GPCR jobs on GPCRDB. But I'm crossing my fingers we can deliver on that promise.
That'd be great. So that's another reason to go take a look at GPCRDB if you haven't done so far. Fantastic. Thank you so much, David, for your time. I really appreciated our conversation. And we'll talk to you soon.
Bye-bye.
Bye. Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Don't forget to subscribe to the Dr. GPCR newsletter at drgpcr.com/newsletter. You can also leave us a 5-star review on Apple Podcast if you think we deserve it, and share our podcast with your colleagues. Until next time, stay safe.